Hi, my name's John Downing, and I'm a limnologist and aquatic ecologist. And this is a session in the limnology and aquatic ecology course that I am the author of. This particular session is about primary producers and primary production, and we'll concentrate mostly on algae and sort of phytoplanktonic organisms in this session, but we'll talk a lot more about aquatic macrophytes and other kinds of vegetation when we talk about littoral zones and other shallow water environments in aquatic systems in subsequent sessions. So this one is about the generalities of primary production, but also the primary producers, concentrating on pretty much the algae and cyanobacteria. So algae and primary production. Here we have some beautiful pictures of very amazing rates of primary production in very what we would call eutrophic ecosystems. Now you're seeing a lot of green algae and cyanobacteria growing here. These are often called the grass of lakes and the grass of oceans because they're small organisms that grow pretty fast and can, in many circumstances, dominate primary production. Here are a couple of other views showing different kinds of primary producers suspended in lakes. So the upper right is probably diatoms because they're kind of a brownish yellow color. You see some very brownish color down to the lower left and some quite green and blue-green color to the upper right. Those of you who've been paying attention to these sessions notice some Langmuir cells in the photo at the lower right, and of course you know that those Langmuir cells are windrowing the plankton as they rotate the water. Here's another view of primary production, this time from space, and you're seeing a bloom of phytoplankton along the coast of Louisiana. If you of the United States, here's the peninsula of Florida, and here we have New Orleans in this area here, Lake Pontchartrain, and the Mississippi River flowing down this way, releasing nutrients that allow marine phytoplankton to grow. Also the nutrients flow in here in the Atchafalaya River, and so you're seeing a lot of this growth. This is what the growth of algae can, of course, fuel hypoxia. So algal growth is good, and it has some bad connotations too. Here's another image of the same area showing the growth of algae as the nutrients are supplied to the sea by inland waters. Here's a close-up picture of some algae growth. This is some that I saw in the state of North Dakota. These are floating cyanobacterial blooms. You see in the inset the view from the air and the view very close at hand in the overall image. Here there are a few things that you may already know about primary production in aquatic systems, and you know that anything that lives in aquatic systems needs primary producers. For example, plants or some other primary producers, and they need those things to turn the sun's energy into food. Now, in some ecosystems, of course, aquatic ecosystems, a lot of food is derived from the landscape that material flows in, but in general, plant production is very important to the production of good food resources for aquatic organisms. Also, if you fish, you know there are big plants out there that you may call weeds, and if you fish, you may be interested in finding those because they supply great habitat for fish. If you swim, you may want to not find them because a lot of people don't like them around their feet and they find them sort of creepy. But we don't call them weeds. We call them aquatic macrophytes. They're another kind of primary producer. But this is something you may already know, that in lakes there are plants, and some of them can be really positive, and they can cause you some concerns as well. If you've ever looked at water closely in a lake or pond, you may see green or brown stuff growing on the bottom or suspended in the water. This is algae. Algae or other cyanobacteria. It can also be the brown stuff, can also be sediments, but quite frequently, if it's green or greeny brown color or blue-green, then you're looking at some kind of phytoplankton that's growing there. And these are small cells usually, and we'll have a look at those. You may also have heard that sometimes there's so much stuff growing in the water that it becomes toxic. And down at the lower right, you can see an image of a warning sign on a beach telling people to stay away from the water if it is particularly green or brown, because it can, in fact, cause health problems to people. So these may be things that you already know about primary production in aquatic systems, and we'll try and build on those ideas. My objectives for session 13 are several. First, I'd like us to review what primary production is, so we can remember what that process is doing. Then I'd like to do a sort of who's who of the primary producers and walk through them, have a look at them, have you think about them, and then I'd like us to discuss the factors that alter primary production, and then think about how primary producers and primary production differ among lakes or among other aquatic ecosystems, and then talk about the seasonality of primary producers, because this is a very obvious thing that one sees as a limnologist, how these primary producers change over season, especially in a temperate area where seasonality is very pronounced. This is the process of photosynthesis, and it's something you've probably known about since elementary school, but I'd like to remind you about it once more. Over here on the left, you see CO2, carbon dioxide, and here is water, and obviously in aquatic ecosystems there's plenty of water. But what photosynthesis is the combining together of those carbon molecules into a more complex, energy-rich compound. And here we're seeing sugar as a result. And this is so we're taking 6 carbon dioxides and making 1 sugar molecule. This is a glucose molecule. And we need these hydrogens here to build this sugar, and so we're liberating quite a lot of oxygen. Interesting thing that we should think about and keep in mind throughout the rest of these sessions is this reciprocal relationship between carbon dioxide and oxygen. Those things that use CO2 tend to give off oxygen. Those things that use oxygen tend to give off CO2. But this whole process of primary production is facilitated by these guys here, by various kinds of primary producers in aquatic systems. They have photoreceptors. In the presence of light, they can combine CO2 and water together to build more complicated, energy-rich compounds that can be used by other trophic levels. So, primary production is the rate at which carbon is fixed by photosynthesis. By fixed, I mean turned into something else, some other complex material. It's usually measured in grams of carbon produced or fixed. It can also be measured by grams of oxygen fixed. It also can be measured as kilocalories of energy fixed. And you can get the sort of reciprocity amongst these simply by looking at this equation. And normally when we express this, we express it in carbon, oxygen, or kilocalories per area of lake or volume of water. And that's a way for us to measure primary production. Now, one thing that we should remember is primary production is usually proportional to biomass, or how much primary producer we have there. And as a measure of biomass, we'll use cell volume, sometimes aquatic macrophyte or water weed sorts of volumes, and chlorophyll concentrations as an estimator of the overall photosynthesizing biomass. Now, the production that's generated within a lake is called autochthonous production, and that's an important word. Autochthonous production is that is auto, it is built within the system, and that's generated outside a lake and imported is called allochthonous or allochthonous outside production. Plankton primary producers in aquatic environments are many, but one of the most important ones is phytoplankton, and this is called the grass of lakes and oceans. And these are small, free-floating algae usually. Some of them are attached to surfaces, of course. but these can be single-celled or colonial, and we'll see quite a lot of them. Also, there are some bacteria that make up the phytoplankton. The two kinds of plankton the phytoplankton are divided up into two kinds of plankton, and those would be the holoplankton and the meroplankton. The holoplankton are those perennial species that are usually present throughout the year, often at very low abundance. The meroplankton are those intermittent species that often organisms with resting stages in the sediments. They take longer to grow when conditions change, but tend to make up a large amount of the plankton and tend to be seasonal in nature. There are different size groupings of plankton, and some of the fonts here may be a little bit hard for you to see on a video screen, depending on how big your screen is. But are you looking down the left-hand column? These are our groupings of plankton. We talk about femtoplankton, picoplankton, nanoplankton, microplankton, mesoplankton, and macroplankton. And these different groupings are based on sizes. So femtoplankton are extremely small. Just as a point of reference, and something maybe for you to jot down and remember, a sheet of paper is about 100 microns thick. So a femtoplankton is usually less than 0.2 microns, which would be really, really small. It's a 500th really of the thickness of a piece of paper, so really quite tiny. These are made up mostly of small bacteria and viruses, and they grow very fast. Now, if you go to the upper scale in the plankton, sometimes you will see very large things, plants free floating. These can be things like duckweed and water hyacinth, various planktonic primary producers, and they can be greater than 20,000 microns. So that is up to maybe 20 millimeters or 2 centimeters in size. So these are quite big sorts of things. Now these, one thing I'd like to show you is, and another thing I'd like you to tuck away as a fact to remember, and one that'll be quite important here, is that little things do things faster. They grow very, very quickly. Big things grow very slowly. So along this size scale, we have a growth rate gradient as well as a size gradient. So the femtoplankton are made up of small bacteria and viruses. The picoplankton are 0.2 to 2 microns, and they're the smallest phytoplankton, and they're quite fast growing because they're very small. The nanoplankton, we call them nano because we used to think they're very tiny until we started doing better microscopy. These will be things like flagellates, and they can flagellates and other kinds of cells, and they're quite common. And they'll be the principal food of zooplankton, that is to say, plankton-eating animals. They grow fast too, but not as fast as the tinier things. The microplankton are bigger cells and colonies, and we used to call these the net plankton, not because it's net versus gross and net. It's because that's what we used to clog. They used to clog up our nets when we drag nets through the water, and they're from about 30 microns to about the thickness of 2 sheets of paper, something like that. So they're pretty small also, but they grow at moderate rates compared to the smaller organisms. And next comes the mesoplankton, and these are larger cells and colonies, and they grow much more slowly. And these are fairly large organisms and colonies, and take longer to explode into bloom. Now there are some kinds of organisms that are small cells that can grow very rapidly and form colonies, and those have sort of they have the strategy of being large, so they're not eaten, but they also have the strategy of being able to grow rapidly. Now you see 2 other columns here in the size grouping. This over here first is a sinking rate. Little things, I said little things do things faster, big things do things more slowly. One thing they don't do, little things, very fast, is to sink. Because they're small particles, they have a high surface to volume ratio. But large things tend to sink rather faster. And of course, because macroplankton would tend to sink very quickly, they often have buoyancy adaptations. Otherwise, they would simply sink to the bottom and could no longer be planktonic. There's also a scale of who eats whom on this. So the femtoplankton are usually eaten by very small things like ciliates and protozoans. Picoplankton are eaten by protozoa, rotifers, and small cladocera. I think you can see here that as we get bigger, the cells get bigger. The things that eat them get bigger as well. The nanoplankton are eaten by rotifers, cladocera, and copepods. Microplankton are eaten by pelagic and benthic fish and invertebrates. Mesoplankton by pelagic and benthic fish and invertebrates also. And then the macroplankton are not eaten by very much of anything at all, except when they're decomposing. And then they offer a supply of bacteria and organic matter for detritivorous organisms to eat. There are also non-planktonic primary producers that I've alluded to earlier, and we'll talk about them in greater detail later when we have a session on the littoral zone. But first we'll just kind of go down this list. There's the neuston and epineuston, and these are planktonic primary producers that live in the surface film. And the surface film that we saw earlier was possible because of the high surface tension of water. Now, there are going to be a bunch of epis following. We have epiphyton, epilithon, epipelon, and episammon. Epiphyton, epi means things that grow on the surfaces of. So epiphyton means grows on the surfaces of plants. Epilithon means grows on the surfaces of rocks. Epipelon means grows on the surfaces of mud. And episammon means grows on the surfaces of sand. So those are the epitypes. There are lots of kinds of primary producers that live on surfaces, and these will be a variety of different kinds of organisms. Things like, of course, the diatoms that we'll talk about in a little bit, but all kinds of other organisms will live there too. Finally, in this list are the aquatic macrophytes, the things that you may call water weeds, and I hope that you won't do that anymore, because they aren't weeds at all. They're extremely useful. Well, there are some that are problematic. They're invasive aquatic macrophytes that are problematic. And these can be vascular or non-vascular plants attached to the bottom. And vascular means having some kind of tissue for conducting gases, and non-vascular do not have the kinds of vascular tissues. So aquatic macrophytes, and again, we'll pick those up later in future sessions. Now we're going to talk about mostly the algae for a while, and these would be the things that we would find oftentimes in the phytoplankton. And the first group of algae we'll talk about will be the first group of algae in this list anyway are the Cyanobacteria. And these are what we used to call blue-green algae. They in fact aren't algae at all. These are much more like, they're prokaryotic organisms like bacteria. So in fact they are cyanobacteria, meaning they are blue bacteria. The blueness is given to them by the pigments that provide photosynthesis. Another one we'll look at is the green algae, then the yellow-green algae, the golden-brown algae, the diatoms, the cryptomonads, the dinoflagellates, and the euglenoids, and the brown and red algae that we don't see very much in freshwater systems. So we'll just kind of step through them one by one. So here we have the cyanobacteria. These I always call the phytoplankton you love to hate, because they are often very toxic and problematic. And when we're managing aquatic ecosystems for problems of eutrophication, they're the ones that cause us the most difficulty. These are prokaryotic cells, and they're lacking many of the membranous structures you may be familiar with, and this would be things like nuclear membranes and chloroplasts and so on. You can see from the image that they have kind of a granular, coloration has sort of a granular aspect to it. So that's because the pigments, photosynthetic pigments, are distributed throughout. the cells not being isolated within chloroplasts. They contain some chlorophyll, like the so-called higher plants, and but the main pigments are bilioproteins, and we saw earlier when we talked about light and light absorption, that the bilioproteins have some special light absorbing characteristics. The cyanobacteria can be unicellular, they can be filamentous, they can be colonial, and they're usually enclosed in a mucilaginous sheath. Try and say that fast 10 times. Mucilaginous sheath can become problematic all by itself because it's sort of a mucoid material, and if they grow in profusion, they make kind of a terrible slime, especially when they're floating on the surfaces of lakes. Now, one of the things that cyanobacteria can do, at least some of them can do, is to fix nitrogen in special cells called heterocysts. What does fix nitrogen mean? Well, again, we'll talk about this in subsequent sessions when we talk about nitrogen and phosphorus. But fixation of nitrogen, I mean, you may know that you're swimming in greater than 90% nitrogen gas, and so, but they can't. But most plants can't use it in that format. So these little heterocysts can absorb N2 gas and turn it into usable forms of nitrogen that the cyanobacteria can use for growth. Now this means that they have some special advantages. They tend to be able to grow when there is very little nitrogen, or when the nitrogen to phosphorus ratio is lower than cells normally need. Another thing they do is they form stinking and toxic algal blooms, and we've talked about that a little bit. These sort of toxic algal blooms can be health hazards to domestic animals as well as people if they get into the water supply or if people come in contact with them. So that's why I say they're kind of the phytoplankton we love to hate. The next group of algae I'd like to talk about are the Chlorophyta, and these, as you can see on the right, are pretty classic. You've probably run across these in middle school or somewhere. Science teachers like to have students look at these because they're so beautiful, and the chloroplasts are often shaped in various beautiful ways, and the coloration of them is often extraordinarily green and beautiful to look at. This is an extremely large and diverse order, and almost all of the green algae, or the Chlorophyta, are almost all freshwater. There are some types of that are flagellated or amoeboid. There's some things called desmids that move in a flowing motion. But the Chlorophyta contain the classic freshwater algae, things like Spirogyra, Eugonium, and a variety of other kinds of beautiful algae. The chlorophylls are the dominant pigments, as you can see from their very green color, and the chlorophylls are contained in chloroplasts, unlike the cyanobacteria. The Chlorophyta also contain some macrophytic algae, and that includes Chara and Nutella. If you have spent much time in northern or temperate water lakes, you probably have run across one of those. Chara is often called skunkwort because it stinks a bit. It has a bit of sulfurous smell when you pull it out of the water. They're very healthy kinds of plants, and there are also some very important and interesting mobile and diatom-like desmids. So a very diverse. The Chara is shown down at lower right, and that's a macroscopic view. That's not a microscopic view. And so you can see that it's a fairly large plant. Now this doesn't have vascular tissue. It's definitely an alga. Next, I'd like to talk about the Xanthophyceae. These are the yellow-green algae, and these are sometimes pests in swimming pools and so on, and people work pretty hard to get rid of them. These are unicellular, colonial, or filamentous algae, and they have conspicuous amounts of carotenoids. And if you think back to when we were talking about light, carotenoids are those sort of carrot or yellowy orange colored pigments, and this is why these algae are called the yellow-green algae. The cell wall is often absent, and they're quite rich in pectin or silica. The cell walls are when they're present. Pectin is that jelly-like stuff that you make. If you've ever made jelly, that's what you've used to gel it. There are a few planktonic members, but most live on sediments or plant surfaces. So they can be epiphytic, and they can be epipellic. Now here you see a couple of different names of the various genera. Here's Rivularia that becomes quite abundant in very acid lakes and so on. Next, I'd like us to look at the Chrysophyceae. These are the golden brown algae. They have they have chromatophores that are very rich in keratin and xanthophylls, and this gives them this golden brown kind of color. And you can see one of them down at the lower right here, sort of gold color. This is Dinobrion. They're mostly unicellular, or they form colonies, and most lack a cell wall, but some are covered in delicate siliceous or calcareous plates. Okay, siliceous meaning made out of glass, basically silicon oxides, and calcareous meaning made out of some kind of calcium carbonate kind of plates. They're often quite small in size, and they might be important constituents of the nanoplankton in some aquatic systems. Some species, like Dinobrion that you see down there at right, are able to thrive in extremely low concentrations of phosphorus. They also function pretty well in high concentrations, but we often find them in very low phosphorus conditions. Next, I want to show you some diatoms. Back in the early 1900s, people were so enamored with microscopy that a very common parlor art was to have a microscope and then slides with colored diatoms placed on them in various patterns that people would find very attractive. Here's a plate taken from a photograph taken from one of those, and there are kind of a lot was made out of diatoms. They're quite beautiful, and we'll find out why. Here's what they look like if you look at them under scanning electron microscopy. Now, those are those cases that they're encased in glass, essentially. So they are quite literally living in glass houses. So there are various kinds: Tabularia, Coconea, Stephanodiscus, Melocyra, Gomphanema, different genera of those planktonic, and they can be epiphytic and epipellic also. But these are the diatoms. Very beautifully sculptured cell walls made out of glass. So they're one of the most important groups of freshwater algae, and you wouldn't think so since they're covered in glass that they might be kind of difficult to eat. But they're very nutrient rich, and often very rich in various sorts of fats and lipids and so on. They have this silicified frustule, and that's the cell wall. There's some planktonic forms, but many of them also are attached to substrates. They can be epi this and epi that. They're also unicellular and colonial forms. Melocyra, for example, you saw in the previous slide, makes a long sort of filamentous forms. There are two main groups, and these are called the centrales and the pinnales. Centric diatoms, and the centric diatoms have radial symmetry, whereas the pinnate diatoms do not necessarily. In pinnales, there are two groups, the biraphids and monoraphid pinnate diatoms, that are almost exclusively littoral, and therefore they can be really diagnostic if you're looking at sediments. These are deposited in sediments and used frequently in paleolimnological analyses of the history of lakes, because they're so well preserved and because different kinds of diatoms live in different kinds of environments. The next group I want to look at, and here I make a little joke in my class, these are the naked flagellates, and because they're naked flagellates, I show them with their eye spot covered so that you can't identify them. But anyway, it's a kind of a poor joke, I suppose. Here you can see them totally nude. So here we have the Cryptophyceae, cryptomonads, naked flagellates, and see one down at the lower right-hand corner in full color. These have no cell wall, ergo naked. They're mostly motile unicells, And they can be very small planktonic algae, and I think I'm going to show you a little video in just a second of these things. They can form dense populations, and often even in very cold periods under very low light. They seem to be quite effective at capturing small amounts of light. They contain a diversity of pigments, giving them apparent colors of olive, olive green, sort of blue, red, or brown, depending on which of the pigments they contain. So here we have a cryptomonad, and this is a special kind of microscopy, and you could look it up on YouTube if you'd like. But here you'll see this moving around by flagellar motion, and they can move quite effectively. See the flagella in what I would call the back end there as it moves about. Quite fantastic, and are supposed to be able to migrate pretty well. Next, let's look at the Dinophyceae. These are the Dinoflagellates, and I think of them sort of as the space scooter kind of phytoplankton. They kind of look like various forms of Star Wars kinds of vehicles. But these are most motile, and they're unicellular algae. Most of them have a very conspicuous cell wall that you can see in that image, and sometimes it's sculptured or bearing spines, so it can be a very substantially sort of formed. The flagella are contained in channels on the cell surface, and you'll see that in just a second because I'm going to show you a very much too long video of Serratium, and we'll sort of cut it short, but it's kind of fun to watch. They form resting cysts that can remain dormant for long periods and then regenerate when mixed back into the planktonic environment. And these are widely tolerant of chemical and physical conditions. In fact, we often find these in lakes or ponds that have been treated with various kinds of herbicides, especially copper sulfate, because they are quite resistant. In fact, you don't really want to have a lot of these. They can be toxic, and they certainly aren't terribly ingestible and usable by various kinds of zooplankton, or they can be problematic. But they're widely tolerant, and by trying to poison out aquatic plants like macrophytes, then we often create another problem, which is kind of bad dinoflagellate plankton. Some of the species, like Serratium, shown there at lower right, change shape with water temperature, and they get more and more sculptured when sinking would be more rapid. And it's thought to be an adaptation to keep them from sinking out of the illuminated environment near the surface. And sometimes these create toxins. Some of the dinoflagellates are responsible in marine systems for things like red tides and so on, well, various kinds of toxic algal blooms. Now here's an image of Serratium moving around, and I think you'll be able to see the flagella in the little channels as they go in quite closely. And I hope you enjoy the lovely music that comes along with this video. If you look in that channel, you can see the flagella. Next, I'd like us to look at the Euglenophytes, the Euglenophysceae, and these are also pretty mobile, and you can see them buzzing around. They have a high biodiversity, but there are very few truly planktonic forms. They're often common in ponds, and one of the reasons for it is that they're really very fragile. They can photosynthesize, but they're also phagotrophic, meaning that they can ingest particles. So these are very versatile organisms. Indeed, they can photosynthesize and also consume small particles. They can also take up dissolved organic matter directly and use that as an energy source. And they can be used as indicators of high concentrations of dissolved organic matter, as they do extremely well when DOC is present. Now here are some euglenoid motion. This is Euglena viridis. The red spotted would be a light sensing organ. They can actually move kind of rapidly. These are taking it easy. Where's there to go, really? And you see some of them sort of bumbling around. But they actually do have some quite good sensory ability for the very tiny organisms that they are. Well, there are some other groups of algae also in aquatic systems, and these are the two that we really haven't talked about are the phaeophytes and the rhodophytes. And phaeophytes are the brown algae. These are mostly marine filamentous or thalloid forms, things like fucus and so on. But there are some freshwater brown algae that are attached to substrates. And then the rhodophyta are the red ones you see there at the bottom. They're mostly marine, but there are a few thalloid or filamentous species found in fast-flowing streams. So if you see some kind of red stuff in a stream, it's probably a rhodophyte. They're not very abundant and not major primary producers in aquatic systems in general. Now there are several. We've talked about several characteristics of algae and cyanobacteria, but there are some characteristics that are of adaptive value that we should think about in specific. One adaptation that algae and other phytoplankton have is the pigment type that they use, the content, and the form and function of the photosynthetic apparatus. Some pigments work better at depth or in colored waters, and this is simply because of the way light is changed by the different kinds of waters in which organisms find themselves. And if you are interested in this, you can look back at one of the earlier sessions on light and in limnology or in aquatic ecosystems. Also, different algae and phytoplankton have different nutrient requirements. Some have evolved efficient pathways for getting nutrients. For example, nitrogen fixation is very important. Others have evolved alkaline phosphatases that allow them to cleave phosphate from more complex molecules. And some can use things that others can't. For example, the euglenoids can use dissolved organic carbon. Also, algae have several adaptations concerned with sinking and floating. Clearly, it's important for algae to stay in a lighted environment because they are mostly photosynthetic. So, if they have a shape or size or form that causes them to sink quickly, that's counteradaptive and won't help them out very well because they will quickly sink away from the lighted upper waters. So large spheres sink very fast and smaller things tend to sink more slowly. Also, there are phytoplankton and algae and phytoplankton that have various adaptations to their density or specific gravity. Some have lipids and gas vacuoles that aid in flotation. Others have mucus on the outside that also is very low specific gravity that keeps them at the surface. Another adaptation that's really obvious from the little video bits that I showed you a minute ago would be motility. Flagella and cilia allow locomotion and allow you to keep yourself in a particular place in the photosynthetic environment. Another characteristic, algal characteristic of adaptive value would be anti-predatory adaptations. Now, being very large is a good thing because most of the predators that you might run into in a planktonic environment are quite small. or being extremely small might be an anti-predatory adaptation, although you can be eaten by some very small ciliates and other tiny organisms. Colonial phytoplankton also are not eaten terribly much by herbivores because they're just too big to get a hold of, although I have seen zooplankton take in a filament of algae very much like slurping in a strand of spaghetti. Also, various phytoplankton, as you saw with the Dinoflagellates, have various spines and protrusions that increase their apparent size and also make them more difficult to eat. So these are four algal characteristics of adaptive value that may allow different kinds of phytoplankton to function better or less well in diverse kinds of ecological circumstances. Next thing I'd like to talk about briefly is process limitation. Process limitation is very important in algae because it's important that they will have the proper mix of stoichiometric materials available to them. And so first we'll start with the overall concept of process limitation, and then we can move forward and think about this in terms of the kinds of elements algae need. So process limitation is very much like building a car. This old Ferrari Testarossa diagram here, you can notice if you know anything about cars that to build a car you'd need 4 tires, 2 doors in this case, 1 engine, and a whole bunch of energy. Now you need a few other things, radio knobs and odds and ends like that. Clearly you do need some other things, but this is the basic overall what we call stoichiometry of the construction of a Ferrari Testarossa. Okay, so if we have 6 tires, 4 doors, 2 engines, and a lot of energy, how many cars can we make? Essentially, we can only make 1 car because we don't have enough tires to make 2. That means the process is limited by the concentration of tires that we have available. If we have 8 tires, 2 doors, 2 engines, clearly the process is limited by the availability of doors. We're missing enough doors, we've got enough tires, we've got a couple engines, we've got plenty of energy, but we only have enough doors to make 1 Ferrari. Likewise, and this is getting ridiculous, if you've got 1 engine, then obviously then the process is limited by the stoichiometry of engines, so we say it is engine limited. And in this case, if we only got 2,000 kilowatt hours available of energy to put it together, it doesn't matter how much of the other stuff we have, we're going to run out of energy. So the optimal supply ratio is 4 tires to 2 doors to 1 engine to 2,000 kilowatt hours, and that's what we call the stoichiometry. Having less of any of these than that ratio will cause the process to be limited by that element. So you can only manufacture the amount of a product for which you have sufficient raw materials. This is called von Liebig's law of the minimum, and it says that the resource in shortest supply relative to its needs, the stoichiometric needs, will restrain or limit a process. And the process that we worry about with phytoplankton, of course, is primary production. There's a sort of another side to this, however, and that's Shelford's law of toleration, an old ecological concept that says that the maximum value can also be important because certain processes can only take place within a narrow range of conditions. And you can think about this with respect to yourself or other kinds of organisms, that temperature may in fact be limiting, or some things like ammonium, unionized ammonia can become toxic if there's too much of it. So we can have too much of a good thing also. But in process limitation, we're more interested in the resource in shortest relative supply, and that is the one that limits the process. Now, this is used in a bunch of different ways in limnology. One way is that we use this is to understand what kinds of organisms can grow under various circumstances. But another way we use this is for management, by trying to remove a limiting nutrient so that we can decrease the rate of eutrophication. And we'll come to that concept later on. So this is the real process of photosynthesis, and it's I showed you a really simplified one a little while ago. But in reality, what we're doing is we're putting together a lot more CO2, and we're putting it together with nitrate and phosphate and a bunch of trace elements in the presence of light and energy. And we're creating not just simple sugar, but creating protoplasm and a bunch of evolved oxygen and dispersed energy. Therefore, you can think about the things that can have an effect on how much algae can grow very easily by looking at this, because heat, light, and several elements are essential to primary production. Therefore, we might expect various available nutrients and other elements to be able to limit primary production or contribute to it as sort of the mirror image of it. So the concept of nutrient limitation is that the limiting nutrient is that is in shortest supply relative to composition or the stoichiometry of the phytoplankton cell. And generally we know that that we have about generally we know a lot about the stoichiometry. It's shown down here. This is kind of the general algal stoichiometry for oxygen, carbon, nitrogen, and phosphorus. 4 very, very important elements. So it takes, in order to make an algal cell, it takes a ratio of 276 oxygen, 106 carbon to 16 nitrogen to 1 phosphorus atom. Now, there are a lot of other things that might limit primary production, and you can think about these if you get into really more complicated circumstances, but generally we think about C, N, and P. Oxygen normally is not limiting unless systems have become quite anaerobic. But here are the other kinds of organisms that, other kinds of atoms that can limit primary production and have been shown to in various terrestrial and aquatic systems, and they would be, this is a little mnemonic to help you remember it: carbon, hydrogen, oxygen, phosphorus, potassium, iodine, nitrogen, sulfur, calcium, iron, magnesium, sodium, chloride, silicon, and aluminum. So those are the most common limiting nutrients, but by far in inland waters, phosphorus is probably the number 1 limiting nutrient, and we also work a lot with carbon and nitrogen. So C. Hopkins Cafe, Mighty Good Salt, should allow you to remember those elements. And if you are concerned about diatoms especially, then you might want to see Al or think about silicate as a limiting nutrient, or silicon as a limiting nutrient, or perhaps aluminum. Now, primary production is proportional to biomass. So oftentimes in aquatic systems, when we are analyzing what we call primary production, we're in fact looking at biomass. On this axis you see, well, let's look at the x-axis. This is chlorophyll concentration. This is a measurement of algal biomass, as I outlined before. And this is the photosynthetic rate. And the dark dots are for phytoplankton. The sort of gray squares are aquatic macrophytes, and the little triangles up here are benthic algae. So when the chlorophyll concentration on a per cubic meter basis gets high, so does primary production, in slightly different ways for different kinds of organisms. Light and temperature also influence primary production, as we might expect, and we have seen this in earlier sessions, but this shows the seasonal progression at various latitudes. For equatorial regions, 15 degrees north, 30 degrees, 45 degrees, this would be kind of the temperate zone, and 60 degrees where you're getting almost to the sort of boreal regions, and getting into much colder climates. But what we see here is, on the top part of the panel, this is energy availability and how this oscillates through the year. And clearly in seasonal environments and temperate zones, we'll see quite a good deal of fluctuation in the energy availability, both in the incident at the surface and underwater areas. You see a very abrupt increase in energy, and of course this represents ice out or loss of ice cover in the temperate and northern climates. This is a photoperiod, and it may surprise you that sometimes people think that at the equator the photo days are long and wonderful. They're always 12 hours, so it's a friend of mine lives in northern Brazil, and she says that basically it's light from gets light at 7 in the morning and gets dark at 7 at night, and it's always that way year round. For those of us who live in the temperate zone, we find that the photo period can be extremely long in the summertime and very short in the wintertime, and even more pronounced in the higher latitudes. Temperature fluctuation, therefore, temperature fluctuation is temperature is high at lower latitudes. It wiggles a little bit as you go a little bit north. It wiggles a lot more as you go north, but overall, the average temperature tends to decline quite abruptly in the northern latitude. And because we know all of these things, including the mixing layers shown here in this graph here, because all of these things vary across latitudes, we can make some guesses as to how primary production, here estimated as milligrams of chlorophyll A per square meter per day, vary across the seasons and in general from overall latitudes. First, very high primary production year-round in the equatorial regions, very low production in the very northern regions, but some peaking with the seasonal maximum in the summertime in northern latitudes to get up to pretty high rates. But the overall primary production is quite low compared to equatorial regions. Here we see long-term fluctuations in phytoplankton biomass for many seasons. This is from Talling's data from Windermere, and you can see these seasonal fluctuations in biomass here as chlorophyll A in that lake, high in the summertime, low in the wintertime, and repeating in a fairly simple pattern over time. Light penetration and latitude are also important in determining primary production. We've discussed this before too, that as we move into the higher latitudes, the incident radiation is overall quite a bit lower, and the angle of that radiation is also has an effect on the photosynthetic capacity within a lake. And so this is so basically at the higher latitudes we have much less photosynthetic capacity, and this is due to light penetration. Nutrients also influence primary production, and I mentioned this before, and we'll take this up in greater depth in future sessions where we're talking about nutrients themselves and also talking about lake management strategies. But nutrient has a great influence on primary production, and generally primary production increases with algal biomass, and chlorophyll A is a good measure of algal biomass. And here what we see on this graph at right is these dots are actually observations. This is the chlorophyll concentration, and this is the summer total phosphorus concentration. Quite clearly, there's an increasing pattern. The more phosphorus available, the more chlorophyll A there is, and the more primary production potential there might be. The community composition varies among lakes of differing mixing and flushing regimes, and this is analyzed in some detail in the text by Jakob Kalf. And here we will see the flushing rates and mixing frequency here, and flushing rates here, and basically those two variables have have a large effect on the kinds of phytoplankton one might see in a lake in general. And where mixing is continuous, we might see with high flushing rates green algae and small diatoms, but with low flushing rates and high nutrient conservation and continuous mixing, we may see some cyanobacteria. Where mixing is daily, again, we may see small flagellates in this case, but moving again to various kinds of cyanobacteria, some of them potentially toxic, things like Cylindrospermopsis and Microcystis, where flushing rates are not large. And finally, if we move all the way up to the right-hand side of this, where there's little or to no mixing, we may see small green algae and chrysophytes with a high flushing rate, but a kind of different kind of phytoplankton, picoplankton, small, tiny blue-green algae or cyanobacteria, some oscillatoria, and various photosynthetic bacteria. So mixing rate and flushing rate have an effect on community composition. Now, in different kinds of lakes of different trophic status, by trophic status I mean oligotrophic, mesotrophic, eutrophic, and hypereutrophic, we may actually find that we get lower primary production overall over the entire water column in lakes that in fact might have more nutrients. And that's simply because here we see graphs of depth where 0 is at the top and greater depth is toward the bottom. This would be primary production profile. Oligotrophic lakes, no high rates, but distributed quite a bit, quite deeply into the system. Whereas contrasting with a hypereutrophic system, where the primary production may become very high, the area under this curve, which is depth times production, which gives us a volumetric overall sort of integral of water column primary production, the area here may in fact be smaller than the area here, or certainly in a mesotrophic or eutrophic lake. And part of the reason for this is that self-shading leads to radically lowered light levels at greater depths in eutrophic lakes. And in fact, if you have a scum of algae at the surface, then chances are you're not going to have much light penetration, and algae won't be able to grow beneath there. Compensation depth is another concept worth learning, and compensation depth is the depth at which gross production equals respiration, or where phytoplankton or primary producers just make enough profit to balance their respiratory demands. Now, clearly, the compensation depth is shallower in eutrophic systems because light penetration is so poor. It's blocked by phytoplankton that keep light from penetrating the water column. And there's a very strong pressure to stay near the surface in hypereutrophic lakes, and this is why you often will see very strong surface scums and algae blooms at the surface in those kinds of lakes. Now, next I'd like to look at the different kinds of phytoplankton that you find in different trophic status of lakes. And on the left-hand side is a graph taken from some of my early work showing how the different kinds of phytoplankton. Here we see Dinoflagellates, cryptophytes, Chrysophyceae, diatoms of Bacillariophyceae, the Chlorophyta and Cyanophyta, vary as a percentage of the phytoplankton biomass across a range of phosphorus concentrations. We've already learned the concept, I hope, in previous slides, that the more total phosphorus we have, the more algal biomass we see, and the higher the primary production. This is a fundamental concept of limnology. Now, in some places there may be nitrogen limitation, but mostly we see phosphorus limitation. Now, when we move to very high phosphorus concentrations, there's really only one kind of organism we see there, and those are the Cyanophyta, the Cyanobacteria, or the blue-green algae. Now, this means that when we have very high concentrations of, say, phosphorus, of this limiting nutrient, we may, in fact, generate a large biomass of a very toxic algal growth. In contrast, when we have very low phosphorus concentrations way down at this end, we see a very much more balanced ecosystem. Yes, sometimes with the cyanobacteria, but with a great deal of the diatoms. You can see diatoms make up a substantial portion of the phytoplankton, even at fairly mesotrophic and lower eutrophic kinds of levels of phosphorus. We have a very balanced, much more balanced community structure when phosphorus concentrations are low. Looked at another way, here's the total phytoplankton biomass in a lake, and here we have the cyanobacteria biomass, and the lines that you see here are show 1% cyanobacteria, 10%, and 100% cyanobacteria. And what we see here is when phytoplankton biomass gets quite high, and you get this sort of self-shading and very high, rich nutrient concentrations, almost all of the phytoplankton, 100% of the phytoplankton, will be made up of the cyanobacteria, same as was shown in this previous image. This means that as phosphorus increases, the risk of cyanobacteria dominance and dominance by those kinds of organisms that can cause toxins, bad odors and taste to the water, and cause health problems. As we become more eutrophic, generally to the right in all of these graphs, the red line here shows the risk of having greater than 50% cyanobacteria in the phytoplankton. The risk always increases, and increases best with the amount of chlorophyll A, or the phytoplankton biomass increases the most reliably. So generally, if we want to not have toxic algae blooms, we want to have low biomasses of algae. And the way we get that is by limiting the amount of phosphorus that you find in that ecosystem. Bloom formation is an important problem that is studied by a lot of people, but you can see in this image from the newer version of the Calf book that can be a surface scum formed on eutrophic lakes, and this depends really on a cycle of growth of the cyanobacteria. So when we're near optimum light intensity and CO2 concentrations, we and the increase and nutrients increase, we create cells, or the cyanobacteria create cells where the gas vacuoles are diluted by increased speed of cell growth. When that happens, the gas vacuoles and mucus doesn't allow them to stay suspended in the upper part of the water column, and the algae tend to sink. They drop into low light intensity, and they don't grow very much. And under high nutrients, they will consume the ballast and create CO2, generate CO2. Under low nutrients, there will be low vesicle gas pressure. This all leads to reduced excess density, and buoyancy can be gained, and gained very quickly, and they can float back up and form surface scums. And so these surface scums are very noxious, and as you know, and you may have experienced this, they cause decreased value of water in many different ways. Energy cycling also varies with trophic status, and again across a phosphorus gradient, because phosphorus is our principal limiting nutrient. Across this gradient, if we calculate the percent total biomass that is made up of edible phytoplankton, that is edible by herbivores in a lake or pond or river or stream, we see that the edible fraction declines pretty rapidly and is declining quickly by the time we get up to about 100 micrograms per liter of total phosphorus. The inedible phytoplankton begin to increase greatly, and as a reciprocal, actually, obviously logically necessary. But the inedible phytoplankton will make up about 80% of the phytoplankton above about 80, above 70, 50 to 80 micrograms per liter. And that means that there's not much for herbivores to eat, or there may be a lot of production, but there really isn't a lot of quality food. The other thing that happens as total phosphorus increases is we change the size structure of the phytoplankton, and when we're in a very low nutrient regime, much of the phytoplankton is made up of picoplankton. If you look back on the chart that we talked about plankton earlier, these are fairly usable by a variety of different kinds of organisms, and as nutrient concentrations increase, then we get also a decreasing picoplankton fraction and an increasing in microphytoplankton biomass as a percentage of the overall biomass. And that means, again, they're often too big to eat by organisms that form food for fish and other trophic levels, higher trophic levels. The next thing I'd like to talk to you briefly about is what the generalized production profiles look like. And we've really seen this already, because here we're on a gradient of, here we have a primary production, so we have we have different ranges of primary production on the x-axis. So this is high primary production. This is depth on the y-axis, with this being quite shallow. We can see a profile of primary production. Sometimes we'll see a profile that looks like this, and this means that there's light saturation in surface waters because of this straight part of the curve. There is no photoinhibition because you don't see a loss of production in the surface waters, and production is decreasing generally due to light reduction at depth. In this particular curve, you see a similar sort of relationship, except for there is severe surface photoinhibition, and this may be due to high ultraviolet exposure. In this case, where we have, this is probably a hypereutrophic lake, we have very high photosynthetic rates in the surface waters, but a very rapid decline. And again, you can see here that sometimes the integral of photosynthesis, the area under this curve, can be actually less than the area under this one, which is more typical of a low nutrient ecosystem. And here we see some very odd sorts of primary production. This final primary production curve usually is because, and it shows that the photosynthetic maximum is in the metalimnion or hypolimnion. Usually we'll see this in a system where nutrients are depleted in the upper waters and where nutrients are leaking out of the nutrient-rich hypolimnion. We get this in clear waters where the hypolimnion is the nutrient source. The photosynthetic maximum is in the metalimnion or hypolimnion, and sometimes this can cause a regeneration of gases like oxygen, for example, especially oxygen, in the hypolimnion, that can rejuvenate oxygen supplies in that area if the waters are very clear and the nutrient source is the hypolimnion. The final thing I'd like to do is to walk through something I'd like you to be able to reproduce and think about, and you're probably best to look at this in your book, but this is a diagram from Horn and Goldman. But basically this is how, in a temperate zone system, different kinds, how phytoplankton succeeds, and gives rise, and it will basically show you the why and the underlying reasons beneath seasonal variation in phytoplankton community structure and biomass. And first we can kind of look at all cells, and then we'll look at the kinds of cells. In the upper panel of this graph, well, clearly the x-axis is season, so we're moving from winter, spring, summer to autumn, and winter again as we move from left to right. And so you can see, if you look at the third panel down, you see something very clearly happening, that there are two big peaks in primary production. These peaks in primary production occur usually at ice out, or when in spring when temperatures increase and solar radiation increases, and sometimes there's a supply of nutrients that comes from the hypolimnion. So you'll get this, and so you'll see these two big humps in primary production. You see them because of increased amounts of radiation and increased nutrients at various times, these 2 times, pivotal times of the year. And the reason you get increased nutrients in spring and fall is because of seasonal mixing, and this would be very typical of what is called a dimictic lake. So the top part of the graph shows solar radiation and heating, and the dashed line is surface water temperature, and then the dotted line in the upper part of the graph is evaporative cooling. So we can see that this the system is warming is heating quite rapidly in the early part of the year, and that heats up the water to some kind of maximum at the end of summertime, and then there's cooling and temperatures tail off. So that tells us that there's a bigger supply of energy in 2 different ways: radiative temperature energy, and also the photosynthetically active radiation becomes abundant in the spring, and that causes a peak in production of phytoplankton in general. Now, if you look down to the bottom part of this graph, this shows zooplankton grazing rates, and these are herbivores that we'll look at in one of the next sessions. But herbivores eat those cells, and you can see the abundance of herbivores. As soon as there gets to be food to eat, then those herbivores graze down those cells, graze them down very hard, so that in fact the overall abundance of those cells tends to decrease through summer to a sort of a summer clear water period. They're no longer blocking a lot of light. All the time that these different kinds of various herbivorous zooplankton are feeding on them. Now you can imagine, however, that if the predatory zooplankton then would grow in abundance, then they would eat these herbivorous zooplankton, and you might see some other peaks in algal abundance. Now, the final thing that we haven't looked at on this plot are these various peaks that are making up the phytoplankton composition. And in springtime, these tend to be individual species of diatoms mostly. They're growing in fairly nutrient-rich circumstances, and probably have substantial amounts of nitrogen to grow on. During the summertime, of course, when the phytoplankton are being grazed down by the herbivores, the kinds of organisms that are there would be small flagellates, too small really to eat, and blue-green or cyanobacterial cells that are not really edible to the zooplankton. So they are there in small abundance. Then what happens is that we get a beginning influx of new nutrients and a release from predation to all intents and purposes, and we get a growth of algae again in the autumn, a very big peak, but these peaks tend to be made up of some diatoms as in spring, but mostly blue-green algae and dinoflagellates, more resistant kinds of organisms, and those with less stringent nutrient requirements, and those that have been able to make it through the summer due to lower grazing rates. So high growth rates only occur after stratification, because after stratification is lost, because cells circulate into the dark before stratification. Stratification cuts off hypolimnetic nutrients. The peaks of algal taxa arise due to different nutrient needs and susceptibility to grazing. Then the rising temperatures increase loss to herbivores, because the herbivores can become more active when it's quite warm. And then the decline of the spring bloom is due to grazing and nutrient depletion in the upper water column. Summer stagnation sets in. This is dominated by algae that are protected from grazing and adapted to live in relatively low nutrient conditions. And then because of the high grazing rates throughout the summer, the fall bloom is often dominated by cyanobacteria and algae that are harder to eat. So here's a summary of this discussion of primary producers in aquatic systems. So first of all, primary production is a process through which sun's energy is fixed by plants and then used. This autochthonous production can be used by organisms within an aquatic ecosystem. This primary production is done by many different groups of algae that we reviewed, and you should probably know something about. And it's also done by bacteria and vascular plants that are free-floating or attached to substrates. Primary production varies, of course, with all those things that can limit it, like nutrients, of course, light, temperature, and various kinds of trace elements and elemental composition of the water, although the principal limiting nutrients are mostly phosphorus, sometimes nitrogen, and in some rare cases, carbon. Phytoplankton communities vary along a trophic gradient from very biodiverse at low nutrient concentrations to cyanobacteria dominated in eutrophic and hypereutrophic ecosystems, and this can be a problem that needs to be solved by lake management. Seasonal variation is driven by a variety of factors, all of those kinds of things that limit the abundance of phytoplankton, but the seasonal variation is driven by the energy inputs, of course, by nutrient dynamics, and also by competition and by grazing.